Hello students, and welcome to Lecture 10 of Human Biology. Today we're going to be talking about the human immune system. More information can be found out about the human immune system in Chapter 42 of the textbook. And today I'm going to start by making a very brief discussion of the types of things that can invade the human body, and these are the things that the immune system typically has to deal with. So the immune system is there to prevent your body from being invaded by foreign organisms, usually microorganisms, bacteria, fungi, viruses, protists, obviously, in case you don't know, not all microbes are bad for us. In fact, only about 5% of the different microbial species in the world are actually bad for us. The other 95% are doing either neutral or are benefiting us. Don't get the common misconception, don't get the idea that microbes are bad for us per se. There's only about 5% of the different types of microbes in the world are actually bad for us. And the ones that are bad for us, we call them pathogens. So the purpose of the human immune system is to destroy invading pathogenic microbes, as well as destroying cancer cells. So there are many people who develop cancer cells, but they don't actually get cancer because their immune system will destroy these cells. The cancer cell basically is just a regular human cell that is malfunctioning so that it doesn't know when to stop dividing. Normally, cell division, when cells divide to make new cells, that process of cell division is normally carefully controlled so that cells in the human body will only divide to make new cells when they're told to do so by the endocrine system or by parts of the endocrine system. So a cancer cell is a cell that has had a mutation, sustained a mutation to one of the genes that controls cell division. That's all. So it keeps on dividing even though it wasn't told to divide. It just keeps on dividing and never knows when to stop, and it ends up forming tumors, these big masses of cancer cells that are just taking up nutrients and blocking blood vessels and blocking other kinds of vessels that cause problems. And so cancer is a very bad thing. It's actually very simple to understand what it is. As I said, it's just a runaway. It's a cell that's sustained a mutation so that it's undergoing runaway cell division. It's dividing and dividing without stopping. And so understanding what causes it is fairly simple. Mutations are random. Sometimes mutations can be caused by chemicals that are called carcinogens. Other things, sunlight, for instance, can cause mutations to DNA and thus run the possibility of causing cancer. But most people have, a few cells here and there that will become cancerous, but they don't actually develop cancer because the immune system destroys those cells. And so that's another function that the immune system has. So I'm just going to briefly talk about what are the different pathogens that the immune system is there to destroy. And then I'm going to move on and discuss the white blood cells and the functions of the white blood cells and how they destroy these invaders. Right, so then we're going to be talking about blood cells for all of these topics. And then at the end, I'm going to spend some time talking about the lymphatic system as a whole. Now, you know that the human body has a network of veins that run to all areas of the body, and it has a network of arteries that run to various areas of the body. So the veins and the arteries are involved with circulating the blood around the body. But you may not know that there's another network of vessels, tiny tubes, vessels, that circulate another fluid around the body called lymph, L-Y-M-P-H, lymph. And those tubes, those vessels, together with a bunch of organs called lymph nodes and some other glands in the body, are collectively called the lymphatic system. So the lymphatic system is another network of vessels, tubes that carries, which are called lymphatic vessels, which carry lymphatic fluid or lymph around the body, and this is part of the immune system, and we'll discuss how that works later on. But just remember, I'm going to talk about white blood cells first, and then after I'm finished with the white blood cells and how the white blood cells destroy invading organisms, I will talk about the lymphatic system and the lymphatic vessels and little things called lymph nodes and how they function in the immune system. All right, so I'm going to start out the discussion on white blood cells with a brief review of hematopoiesis. You may remember from an earlier lecture that I mentioned that hematopoiesis is the formation of blood cells from stem cells that are located, things that are called bone marrow stem cells that are located inside spongy bone, inside the red bone marrow. And hematopoiesis is a very interesting subject. It's a very complicated subject. There are people who spend their entire lives just studying hematopoiesis, and we still don't know very much about it. There are medical doctors who specialize in the formation of blood, and they are called hematologists. So hemato refers to blood, and then logi means to study. So there are medical doctors that have MD degrees that specialize in all aspects of formation of the blood, cancer of the bone marrow, and so on. And those people are called hematologists. Now, it turns out that when you convert red bone marrow cells, when you convert red hematopoietic stem cells into blood cells, they belong to one of two families. Those families are called lineages. Right? So your ancestors are your family line. The word line is the root word of the word lineage, right? Your family line. So blood cells, when they are created and they join one of two lineages. One lineage is called the granulus, or one lineage is called the lymphocytes, and the other one is called the erythrocytes. Okay, so there are actually two. There are other lineage. I'm sorry, I meant granulocytes. So blood cells belong to lineages. There are two broad types of blood cells. Those that are red are called the erythrocytes. Red blood cells are called erythrocytes, and white blood cells are called leukocytes. So as I explained before, erythro is a Greek word for red. Leuko is a Greek word meaning white. Ancient Greek, so if you said those words to a modern Greek, they may not understand, but this is ancient Greek words. And then those leukocytes can be further subdivided into two categories. So the leukocytes can be either granulocytes and lymphocytes, and we'll get into this later, but the two family names for the two lineages are called the myeloid lineage and the lymphoid lineage. Right, so we have two main lineages, the myeloid lineage and the lymphoid lineage. The erythrocytes, the red blood cells, together with the platelets, which we discussed when we discussed the circulatory system, the red blood cells, the erythrocytes, and the platelets are members of what is known as the myeloid lineage. And everybody else is a member of the lymphoid lineage. Right, so we'll talk about that later. Okay, so the granulocytes have a grainy appearance when you look at them under the microscope. So granulocytes have a distinct appearance, which makes them easy to differentiate or to tell apart from lymphocytes. The granulocytes have a grainy appearance, and they are involved in something called the innate immune system, the innate immune response, which is a pre-programmed immune system response, where the immune system is pre-programmed from the day that you're born to attack certain types of microbes. Okay, so we have something, the various jobs that these white blood cells can do are divided into two distinct functions. They do two distinct things. There's one type of cell, the granulocytes, who carry out the innate immune system. Now, if you studied psychology or if you know what the word means from somewhere else, the word innate means that you're born with this ability. It is innate, right? So you should know the word innate. So the word innate means that you're born with the ability to do this, and so we have cells called granulocytes that from the day they are created, they know how to go after and destroy certain microbes, like a bacteria called Streptococcus pyogenes, for instance, or another bacterium called Staphylococcus aureus. We have granulocytes. One of the types of white blood cells, we have granulocytes that will automatically start attacking Staphylococcus aureus or Streptococcus pyogenes. It's part of the innate immune response. But there are other white blood cells that they will attack foreign invaders, but they have to go through a long, complicated process of first learning what those foreign invaders look like. Those cells have to learn what the foreign proteins that are on the surfaces of those invading cells look like. And because they have to adapt to the invader, this is called the adaptive immune response. It's sometimes referred to as the learned immune response. And the cells that carry out the adaptive immune response are the lymphocytes. Right? So the granulocytes and the lymphocytes are both leukocytes, but not all leukocytes are granulocytes, and not all granulocytes are lymphocytes, or not all leukocytes are lymphocytes, for instance. Right? So the granulocytes, which are involved in the innate immune system, and the lymphocytes that are involved in the adaptive immune system, the adaptive immune response, are both classified as leukocytes because they're white. The word leuko simply means white, as opposed to red, the erythrocytes. Okay, so the two types of one type in particular of the lymphocytes is called a B cell. And it is B cells that secrete or produce the antibodies. You might have heard the word antibodies. Antibodies are like little bullets that the B cells fire at the invader to kill them. And antibodies are very specific. They will only attack a very specific foreign protein. So B cells learn what the foreign proteins on these invading cells look like, and then they differentiate. They turn into, the word differentiate in this sense, in the cell biology sense, the word differentiate means to turn into a specialized type of cell. So a B cell is not quite a specialty cell yet. It learns what the foreign proteins on these invading microorganisms look like, and then it differentiates into another type of cell called a plasma cell. And the job of a plasma cell is to make billions and billions of antibodies. And these antibodies then swarm like a bunch of hornets or bees to attack the invading microbes. So I think like antibodies, you can say there's the analogy would be that they're kind of like wasps that are swarming to attack an animal that's attacking their hive, you know. And so the antibodies are like that, except that the antibodies will only specifically attack a very specific foreign invader, and it has had to have taken some time to learn what that invader looks like. But the adaptive immune response ultimately ends up in the production of antibodies. Okay, now the immune system, after the adaptive immune system, after it has learned what the enemy looks like and destroyed the invading cells, it leaves behind some B cells and another type of cell called a T cell that remember what it looked like. So the next time it shows up, they're ready for it. Right? So the next time the second time the invader shows up, the human immune system and the immune system of every other mammal, the immune system is ready to destroy it instantly. And that is called the secondary immune response. And that is, we exploit the primary versus the secondary immune response in order when we make vaccines. Because what a vaccine is you take a virus or you take a bacteria and you kill it, and then you inject it into your body. That's called a vaccine. Your immune system will recognize this foreign invader with the foreign proteins. It will take about 2 weeks to learn to adapt to this, and then it will produce B cells that will produce antibodies that can destroy it. That's referred to as the primary immune response. The first time if you haven't been vaccinated, the first time you're exposed to a virus, you'll get very sick, and it may kill you, depending on which virus or which bacteria it is. But if you survive it, the second time you get the same disease, you will not get anywhere near as sick. You'll probably survive it much better. And so there was a famous scientist named Paul Ehrlich in the 19th century who figured out that why is it that you get hit very hard with a disease the first time you catch it, but every time you catch it thereafter you get less and less sick every time. There must be something in your body that learns how to combat this invader and provide you with immunity, exemption from sickness. Actually, that's what the term immunity means. It means exemption, but in this case it means exemption from sickness. Okay, so we exploit this when we take these kind of pathogenic viruses and bacteria and we kill them and then inject them, so that this time, the first time your immune system sees this bacteria or this virus, it can't hurt you, but nevertheless your body will still mount a primary immune response to it and leave behind memory cells, because the next time you see it, it'll be real, it'll be for real. And so the next time your body sees that virus, it'll be live, but your body will be prepared to kill it instantly. And so that's what a vaccine is. You are exposing your immune system to the virus or the bacteria while it's dead, but you're giving your adaptive immune response a chance to prepare for it for the next time you see it. It's like practicing, right? So, so you learn, you give your immune system a chance to learn what the enemy looks like and to prepare for it. That's what a vaccine is. All right, now sometimes some people have what is known as a hypersensitivity reaction to certain things. It's called an allergic reaction. And that is basically the immune system kind of goes crazy and it causes something called inflammation, an inflammatory response to something like pollen or maybe peanuts if you have a peanut allergy, or bee stings if you have a bee sting allergy. All right, so those are the white blood cells and what they do. We'll talk about those in greater detail, and then after that we'll talk about the lymphatic vessels and the lymph nodes. Okay, I'm just going to give you a very brief introduction to what types of foreign invaders, what type of pathogens can invade the human body. Okay, so there are several different types of organisms on Earth. We talked earlier on about bacteria, which are called prokaryotes versus eukaryotes, and we talked about the fact that eukaryotes can be multicellular or single cellular. A eukaryote that is composed of only one cell is called a protist or a protozoa. A microscopic worm is something called a helminth, which we're not going to get into in this course, but there are other courses for that. And a virus is actually not a living thing. It's a set of rogue genes that travels from cell to cell, usurping the metabolic machinery of these cells. So in other words, a virus is it's a little case that contains some genes. That case is able to get into the cells, get into your cells, and once inside, it takes over the ribosomes and uses the ribosomes and the other machinery in the cell to build more viruses, which then go out and take over more cells. And so a virus is not actually classified as a living thing, but we do study it if we're studying microbiology. Okay, so bacteria, example, Streptococcus pyogenes and Staphylococcus aureus are two examples of pathogenic bacteria that are very bad for us if they get inside the body. You may remember from the digestive system that we have E. coli, Escherichia coli, in our intestine. And as long as the Escherichia coli, the E. coli, is in our colon, it's okay because it is providing us with vitamin K. If E. coli gets into your bloodstream, it is pathogenic, right? So it's not supposed to be there, and it will do great damage to your body if you get E. coli in your blood, right? So part of the what determines whether a bacterium is bad for you or not depends on where it is. So Staphylococcus aureus, for instance, lives on the surface of human skin all the time, and it doesn't do us any harm as long as it's outside the body. But if you cut yourself and the Staphylococcus aureus gets into your blood, the immune system will immediately react and destroy it. Granulocytes will swarm like a bunch of bees to destroy it because it will do great damage if it's inside the body. Okay, so Staphylococcus aureus and Streptococcus pyogenes are two examples of bacteria, prokaryotes, that are pathogenic to the human body. These are pathogens. Some of the things that invade and damage the human body are fungi. Fungi are kind of like yeast. You know what a fungus is when you get like molds, for instance, are members of a family called fungi. One of the most common fungi that infects and damages the human body is called Candida albicans. Candida albicans causes yeast infections in women. So the cause of yeast infections in women is Candida albicans, which normally lives on the surface of the skin. It somehow manages to get into the vagina and causes havoc once it's inside there because it's not supposed to be there. And so you kill it with chemicals that are inside a treatment called monostat. So monostat is a chemical that kills fungi. It's a chemical that specifically kills fungi without killing the person that the fungi is infecting. The same types of fungi have gotten into our fingernails sometimes. These are fungi that eat the dead skin cells, and so they infect the skin under your fingernails and so on. There's another fungus that causes a common ailment called athlete's foot. So athlete's foot is a fungus that attaches itself to the bottom of your feet when you're walking around bare feet in a locker room as an athlete, the way an athlete does. Okay, so athletes are commonly walking around in bare feet in the locker room. So if you walk around in bare feet and there's another athlete in there that's walking around and they have athlete's foot, they will put that fungus all over the floor, and then you step on it with your bare feet, you will get the fungus attacking your feet as well. That is again caused by a fungus. It's caused by another fungus, another type of fungus called Trichophyton rubrum, which we're not going to talk about here. But these are fungal diseases, and a fungal disease is, by the way, a fungal disease is referred to as a mycosis. Mycosis, because the study of fungi is called mycology. Okay, so a mycosis is a fungal disease that infects humans. Examples of protozoa, protists. Okay, so here we have a protist called Plasmodium vivax on one side. On the other side, we have another protist called Trypanosoma brucei. Plasmodium vivax, of course, is carried by mosquitoes, and it causes a disease called malaria. Trypanosoma brucei is carried by tsetse flies, a type of a fly that's found in Central Africa called the tsetse fly, and it causes a disease called sleeping sickness. So malaria, of course, is carried by mosquitoes. There are some other types of parasites that are called helminths, which are microscopic worms. So this is a parasite called Wukariria bancrofti, and it is the cause of a disease called elephantiasis. And so what actually happens, it's kind of interesting that the Wukariria bancrofti worm is just the right size to clog the lymphatic vessels. And we're going to talk about the lymphatic vessels at the end of this lecture. But one of the functions, in addition to the immune system, one of the main functions of the lymphatic system is to reduce swelling. So I mentioned in the last lecture that sometimes plasma will leak out of capillaries into the interstitial spaces of soft tissue, and that causes swelling. And one of the ways to reduce the swelling is to produce more albumin, more serum albumin, to suck some of the water back into the capillaries. But the other method, the backup system for reducing swelling, is the lymphatic ducts that drain away the extra fluid in the interstitial spaces. So another function of the lymphatic vessels is to reduce swelling. If you block those lymphatic vessels with these stupid little worms, the Wukariria bancrofti, if you clog these vessels, you will have uncontrolled swelling. And it often happens to your legs or your arms until your leg or your arm swells up until it looks like an elephant trunk, which was where the term elephantiasis came from. Okay, there are some viruses like rhinoviruses that cause colds. There are influenza viruses. There are measles viruses that cause the measles. Those are not living things. And finally, most people throughout the course of their life will develop a few cancer cells here and there, and the immune system, the white blood cells of the immune system will destroy those cells. So here we have a white blood cell called a macrophage that's eating. It's literally enveloping and digesting a cancer cell to get rid of it. Okay, what about antibiotics? Does anyone know, do you all know what they do? Are you aware of the fact that antibiotics only kill bacteria? So there's no reason to ever go to the doctor for antibiotics. There's no reason to ever go to the doctor and ask for antibiotics. Never a reason to ask the doctor to write you a prescription for antibiotics if you have the flu, because the flu is caused by a virus and antibiotics do not do anything to viruses. They only kill bacteria. So you're only supposed to get antibiotics if you have a bacterial infection, never when you have the flu or influenza, never when you have other things. Some people go to the doctor and ask for antibiotics when they have a cold, and if that doctor is not doing their job properly, they will grant the request and write a prescription. There's a reason There's a reason why in most places in the world antibiotics are prescription drugs. You cannot buy them over the counter at the pharmacist because people will take them for the wrong reason. Now, the reason why that's a problem is because bacteria are building up resistance against antibiotics. So if you don't know, the world was quite a dismal place before the mid-1930s when antibiotics were invented. Antibiotics were invented in the 1930s. Before that, people used to die all the time from bacterial infections. People would if they would get a cut and the cut would get infected, it could actually kill you. And the only recourse that you had was if it was a cut on your hand or something, you might get gangrene. Gangrene is a bacterial infection, and there was nothing you could do to stop it except cut off the arm and hope that you got it all. And if you didn't get it all, it would continue on to the rest of your body and kill you. So before the 1930s, we had a terrible time with people getting sick and dying from bacterial infections. Then when we discovered antibiotics, we had a golden age where we could kill bacteria quite easily. And we, like most things that seem to be really good, we abused them and we overdid it. And now the antibiotics are developing resistance. And so it won't be long if we carry on the way we're carrying on, it won't be long before the antibiotics don't work anymore because all of the bacteria in the world are resistant to them. So that's the reason why you're not supposed to go to the doctor's office and ask for antibiotics when you have a cold. Some people do, because when you take antibiotics, most antibiotics also act as a mild stimulant. So you kind of feel better, you feel a little bit better because you've taken a chemical stimulant, but it actually does nothing to cure the cold, nothing at all. In fact, you would probably feel equally good if you took caffeine pills or if you drank some really strong coffee. You would feel better because it's a stimulant, the same way many antibiotics are stimulants. But the doctor is not supposed to write you a prescription for a cold, not supposed to write you a prescription for antibiotics when you have a cold. If they do, they're being very irresponsible. They're not supposed to do that, because giving constant misuse of antibiotics, giving them in situations where they're not appropriate, is leading to the rise of what are called superbugs. Superbugs are antibiotic resistant bacteria that are resistant to more than one type of antibiotic. So we only have a certain number of different types of antibiotics. If we abuse them by using them improperly, we won't have them anymore. Okay, so antibiotics only kill bacteria. They should never be prescribed or asked for anything else. Okay, where do the immune cells, the white blood cells of the immune system come from? We talked about that already. They're created through a process called hematopoiesis. You must know that word. It's a very important word. And the word hemo refers to blood, or hemato. Right. So hematopoiesis refers to formation of red and white blood cells in the spongy bone. So hematopoiesis, formation of blood cells, both erythrocytes and leukocytes in the spongy bone. The cells that differentiate into the blood cells during the process of hematopoiesis are referred to as hematopoietic stem cells. So make sure that you know that word. Okay, and then we said that we have erythrocytes and leukocytes. The word erythro means red, the word leuko means white. All right, so here is the spongy bone where bone marrow is created. Any of the flat calcaneous bones is where hematopoiesis takes place. Every day, billions of our red blood cells are dying and getting broken down and recycled by the spleen, and therefore every day those red blood cells are being replaced by hematopoiesis, hematopoietic stem cells differentiating into erythrocytes in the spongy bone tissue. All right, now all of the blood cells that are created belong to one of two lineages, and a lineage is a family, comes from the word line. Those two lineages are the myeloid lineage and the lymphoid lineage. Okay, so you do need to know some of the cells that are in each of these. Okay, so the myeloid lineage includes the erythrocytes, the red blood cells. The red blood cells are members of the myeloid lineage. So are the platelets. Remember I talked about platelets in the last lecture. Their function is to cause blood clots and to plug holes, to patch up the holes in damaged blood vessels. That's the function of platelets. Platelets are also members of the myeloid lineage. The lymphoid lineage contains, but is not limited to, it contains the B cells and the T cells that I talked to you about on an earlier slide. Okay, so what causes the hematopoietic stem cells to differentiate into the different cells in the bloodstream? They differentiate, they turn into, differentiation is a cell biology term that means turning from a general cell into a specialized cell. That is called differentiation. And so when a cell does that, you say that that cell differentiates into another type of cell. Okay, so what is it that causes the hematopoietic stem cells to differentiate into red and white blood cells? They do this in response to intercellular signaling by cytokines and interleukins. Okay, cytokines and interleukins are little tiny proteins that one cell sends to another in order to send that cell a signal telling it to do something. So there are some cells in the bone marrow that send cytokines or interleukins to other cells, to hematopoietic stem cells, and telling those cells to differentiate into granulocytes or leukocytes or lymphocytes or erythrocytes. So the word interleukin is kind of handy because inter, you know what inter means, as in inter means between, like in the international airport, and leukin refers to leukocytes. So interleukins are protein signals that are sent between leukocytes, often in the bone marrow, but sometimes outside the bone marrow, signaling them to do things. Okay, so here we have hematopoiesis, and here we have on the right, we have a family tree. So we start out with a generic hematopoietic stem cell, which is constantly dividing to replace itself, right? So it's dividing all the time to replace itself. In response to certain cytokines and interleukins, that hematopoietic stem cell will differentiate into either a member of the myeloid lineage or the lymphoid lineage. Okay, so you can see that if you stimulate this cell with a certain set of cytokines or interleukins, it will differentiate into a myeloid stem cell. And then if you stimulate it with another cytokine that's called EPO, stands for erythropoietin, it will differentiate into a red blood cell, into an erythrocyte. On the other hand, if you stimulate that cell with two other growth factors, interleukins called stem cell factor and totipoetin, I believe that's what it's called, it will differentiate eventually into platelets. On the other hand, if you stimulate that stem cell with interleukin 2, interleukin 7, interleukin 12, and a few others, it will differentiate into either a B cell or a T cell. So you can see this process of turning one general type of cell into a specialized cell is very, very complicated. By the way, just on a personal note, you see this writing here where it says SCF, stem cell factor. I spent 5 years of my life studying just that when I was in my 30s. So I wasted some of my youth studying the proteins of the hematopoietic stem cell system. So anyway, that's one of many interleukins and cytokines that kind of orchestrate the development of hematopoietic stem cells into different specialized blood cells. So here's a close-up again. We have the general undifferentiated hematopoietic stem cell here. If you stimulate it with certain types of cytokines, you can convince it to differentiate into an erythrocyte. Specifically, the important one is erythropoietin, that one there. right? If you stimulate it with another combination over here, notice that we have something called granulocyte macrophage colony stimulating factor. That's what that stands for. Plus interleukin-3, it will differentiate into a granulocyte that's called an eosinophil. An eosinophil is a specialized granulocyte, white blood cell granulocyte, that is specialized to kill microscopic worms like Wukariria bancrofti. It's part of the innate immune system. So your body has eosinophils in it, in your blood, so that if you get invaded, if you get bitten by a stupid mosquito that carries Wukariria bancrofti, elephantiasis, by the way, is carried by mosquitoes as well. Mosquitoes are quite nasty things. So if you get bitten by a mosquito that's carrying Wukariria bancrofti, and that mosquito injects you with some Wukariria bancrofti worms, if you're lucky, your eosinophils will instantly see them and attack it and start destroying them before you get elephantiasis. So most people who get infected with elephantiasis usually don't develop elephantiasis because the eosinophils in their body are quickly sent into action to destroy the Wukariria bancrofti. Okay, so you can see that this is a very complicated process where we have a hematopoietic stem cell which, in response to different combinations of cytokines and interleukins, can differentiate into or turn into any of these other specialized types of white blood cells and red blood cells. And here we have the red blood cells and the platelets. So I mentioned that those are both members of the myeloid lineage. So this whole area over here is the myeloid lineage. The lymphoid lineage is over here. Okay, so the myeloid lineage includes erythrocytes, red blood cells, platelets, and granulocytes. The granulocytes, one example of a granulocyte is an eosinophil which is pre-programmed to kill microscopic worms like Wukariria bancrofti, which causes elephantiasis. The lymphoid lineage includes a type of cell called a T helper cell and a B cell, and I already mentioned that B cells, in response to a certain combination of cytokines, B cells will differentiate into plasma cells, and it is the plasma cells that secrete and produce the antibodies. All right, so we have two types of leukocytes. Remember that leukocytes is simply a term that means white blood cell. So we have granulocytes, which are members of the myeloid lineage. They have a grainy appearance because their cytoplasms, you remember the cytoplasm is the inside part of the cell, their cytoplasms are filled with small storage vesicles. A vessel is something that you use to store things. If you're talking about a microscopic vessel inside a cell, we call it a vesicle. So inside these cells, we have little tiny vesicles that contain things. For example, the eosinophils are an example of a granulocyte. The eosinophils contain vesicles that contain protease enzymes. Do you remember what proteases are? Proteases are enzymes that break down proteins. And so what happens when a microscopic worm gets into the body, the eosinophil will swarm to the area so that these eosinophils will surround this worm, and they will unload all of these proteases onto the worm and start destroying its proteins. So that's how the eosinophils work. There are other granulocytes that work differently because their vesicles contain other things, but that's basically the idea. Okay, now the granulocytes have funny shaped nuclei that are kind of, they might, you remember when I mentioned that if you have an organ that you divide into two pieces, each piece is called a lobe. The same terminology applies to the nucleus. So the nucleus is supposed to be round, but if you have a weird looking cell where the nucleus is kind of divided into two lobes, that's an example of probably an example of a granulocyte. And so because the nuclei have funny shapes and multiple lobes in granulocytes, they are sometimes referred to as polymorphonuclear cells. Poly means many, morpho means shape, and then nuclear means referring to the nucleus. So polymorphonuclear cells are granulocytes, and they can be abbreviated PMN cells. So the PMN cells is another name for the granulocytes. So that's a good question I could ask you on a test. What is a PMN cell? It stands for polymorphonuclear cell, and it is usually it's a granulocyte. So one example would be an eosinophil. So if I said, give me an example of a granulocyte, you can say eosinophil. If I say, give me an example of a polymorphonuclear cell, a PMN cell, you can say eosinophil. Okay, the lymphocytes are also part of the lymphatic system, but they actually get the name lymphatic. So the lymphocytes are members of the lymphoid lineage. They have a round nucleus that takes up most of the cytoplasm, which is the way that you would recognize them. And they're part of the adaptive immune response. The granulocytes carry out the innate immune response when certain types of microorganisms get into your body, and these cells automatically swarm to attack them, versus the adaptive immune response, where the lymphocytes will see an invader and then have to take about 2 weeks to process the information of what it looks like in order to mount an antibody response by B cells specifically that have differentiated into plasma cells. Plasma cells, their main job is to secrete antibodies. Okay, so as I said, the innate immune response is carried out by granulocytes that are pre-programmed to attack certain invading microorganisms. Okay, so as I said, the innate immune response is carried out by granulocytes that are pre-programmed to attack certain invading microorganisms. It is a non-specific response, and I like to use this analogy, and I'm going to use it again in the lecture. The lecture is that if the immune system is like the police department, and the invading microorganisms are like criminals who are coming into your city or your neighborhood, the adaptive immune response is kind of like the neighborhood police, because they're only concerned with catching only the criminals. So their job is to destroy the criminals, and they have to know exactly what they look like. They can't just go around randomly destroying people that might be criminals. At least in theory, they're not supposed to do that. So the adaptive immune response is sort of like the neighborhood police that are looking for a specific criminal. The adaptive immune response will only destroy a specific invading microbe. The granulocytes, on the other hand, are a little bit like the riot police, because the riot police are called in when there's a riot, and these guys show up and they fire tear gas and they start hitting people with their truncheons and making a lot of noise, and they don't care whether you're a criminal or not. Their job is to get you out of there. They have a crowd that has some criminals in it, but they're going to treat the entire crowd as if they're all criminals in order to get them out of the area. That's kind of what the granulocytes do. So granulocytes, as soon as they see that there's a criminal in some area of the body, like Staphylococcus aureus, they swarm to that area and they start damaging the area and hopefully destroying the invader. But in the process of destroying the invader, they cause inflammation. And inflammation is caused by kind of general damage to the tissues in the area. So when you have a cut and it becomes infected, if it gets really nasty, it swells up and it's red and it hurts, that's called inflammation. And that is something that the granulocytes are doing. Right? This is not something the lymphocytes are doing. This is something that the granulocytes are doing, because they're like the riot police that are a little bit too enthusiastic about doing their job. And so I like that analogy. I think it's a good analogy. Okay, so the adaptive immune response is like the neighborhood police. The innate immune response is like the riot police. Okay, let's talk first about the innate immune response. It is mediated by granulocytes. It is controlled and carried out by granulocytes. These are cells that are pre-programmed to recognize and immediately attack foreign invaders like bacteria and protozoa. And they cause something, there's an immune response called the pyogenic response. Now don't confuse the word pyogenic with the word pyrogenic, because the word pyro means fire, and the word pyo refers to pus. Right? Have you ever seen a cut? You get a cut and it gets infected and it gets this yellow, sticky, smelly goo coming out of the cut that we call pus. Pus, right? So pus is actually dead granulocytes, billions and billions of dead granulocytes, and those dead granulocytes died because they were doing battle with the foreign invaders. And the way that they dealt with these foreign invading microbes is they ate them. They ate them and then they died. Right? Basically, that's what happens. So the granulocytes, certain types of granulocytes are specialized to attack and eat and destroy specific foreign microbes, and they know which ones to attack and to eat and to destroy from the day that you're born. So that is the innate immune response. And when you have this response, this formation of pus, it is called the pyogenic response. And if you go on to study medical microbiology or clinical microbiology, if you went into medicine, you would know that there are certain types of bacteria that are classified as pyogenic bacteria. The pyogenic bacteria are the specific bacteria that cause the formation of pus, the specific types of bacteria that provoke the pyogenic immune response. Staphylococcus aureus is an example of a pyogenic bacteria. So if it lives on the outer surface of your skin, if you cut yourself and it gets on the inside, you will get one of these pus reactions. You'll get a reaction, formation of pus on the wound. Okay, now the advantage of this innate immune response is that it's fast. It happens immediately. The disadvantage is that it, like the riot police, it damages law-abiding citizens as well as the criminals. So these cells swarm to the area and they destroy the invader by eating them, but it also has the effect of damaging the healthy tissue that surrounds it. Okay, so the advantage of the innate immune response is that it's fast, but the disadvantage is that it's somewhat non-specific in the sense that it damages the invaders as well as the healthy tissue. Damages the healthy tissue as well as the invaders. And furthermore, the reaction, they cause a reaction called inflammation, which we'll get into in a minute, but it sometimes causes a reaction and inflammation to something that's really not that bad, like pollen, for instance. So people who have hay fever, the granulocytes in their immune system are overreacting to the pollen. They're making a tremendous fuss about the pollen, which is really not very bad for you. It's not terribly bad for you. But it just happens to be something that these granulocytes don't like and they don't react to. They overreact as soon as they see it. And this is the cause of hay fever, which is something called a hypersensitivity reaction. All right, now many of the granulocytes are classified as something called as a class. There is a class of granulocytes called the phagocytes. The word phago is an ancient Greek word meaning to eat. So I mentioned that many of these granulocytes destroy the invading microorganisms by eating them. Once they've eaten them, they often die. Sometimes they don't, but sometimes they do. Okay, so phagocytes are a class of granulocytes, a class of leukocytes. Remember, leukocytes, all white blood cells are leukocytes, but not all leukocytes are not all granulocytes, for instance, are leukocytes, but not all leukocytes are granulocytes. So it's a subclass. Okay, so the phagocytes are a class, a group of the granulocytes whose job it is to eat things, to eat foreign invaders. There are other ones who attack the foreign invaders by releasing enzymes. I mentioned the fact that the eosinophils release proteases onto microscopic worms, so they don't actually try to eat the worms because they're way too big. Instead they dump proteases onto them. They put proteases onto them that destroy their tissues. So here's a phagocyte that's in the process of eating a cancer cell. So that's an example of a phagocyte. This is another type of phagocyte called a macrophage. I showed you this picture before. It is reaching out and spearing and grabbing bacteria. These little oval-shaped things are bacteria. You pull them back. It pulls them back and puts them on the surface of the cell where they are engulfed by phagocytosis. You remember we talked about that in lecture 4, phagocytosis. So they grab hold of these bacteria, they reach out and grab hold of these bacteria like an octopus. They stick the bacteria to their own surface, to their own plasma membrane, and then the plasma membrane engulfs the bacteria through phagocytosis, and it's literally cleaning up your body by eating these bacteria. Okay, the phagocytes are the cells that are classified as phagocytes. Really, they're sort of the same cell. They're a very similar cell, but we give them different names depending on where we find them. So if you find a phagocyte in the blood or in the tissues, it's referred to as a neutrophil. If you find a phagocyte in lymph nodes, which we're going to talk about shortly, it's called a macrophage. If you find phagocytes in other tissues, close to the skin usually, it's called a dendritic cell. Now, these phagocytes, whose job it is to eat things, are also, they are of course part of the innate immune system. That's why we're talking about them now. But they also, some of them also play a critical role in the adaptive immune system, which is mediated by the B cells and T cells. Okay, and they fit into the adaptive immune response because these cells, these phagocytes, eat bacteria and then present those bacteria onto their surface for T helper cells to look at and recognize. So they present the foreign proteins, which are called antigens. They present the foreign proteins to a T helper cell, and therefore they are referred to as antigen presenting cells. Antigen presenting cells. Okay, other granulocytes that do not kill things by engulfing them include eosinophils, basophils, mast cells. Mast cells are an interesting one. Mast cells are involved in the allergic response because mast cells can, the graininess inside a mast cell, the vesicles that are inside a mast cell contain a chemical called histamines. Histamines are a chemical that makes the capillaries permeable, leaky. The word permeable means punch holes in it so that it leaks. So histamines cause the capillaries to become leaky. Now, why would you do that? Well, let's suppose that you had a bacterial infection in your soft tissue. Let's say you've got a bacterial infection inside your adipose tissue, for instance. The white blood cells are in the blood, and the bacteria is in the soft tissues. So what's the point of that? How can the white blood cells get at the bad guys if they're contained in the blood vessels? So in response, the body says, look, the bad guys are out there, the white blood cells are in here, we need to get out there so that we can get at the bad guys. The mast cells will release their histamines, and that will cause the capillaries to become leaky enough that the white blood cells can actually get out. Now, if the capillaries are leaky enough to allow the white blood cells out, you know that the red blood cells are smaller than the white blood cells, and you know that the plasma is not even a cell. So if you have to make the capillaries leaky enough to let the white blood cells out, you know that the red blood cells and the plasma is also going to leak out. So when you let the plasma leak out, you get swelling. And when you let the red blood cells out into the soft tissues, you get redness, the appearance of redness. So that's one of the reasons why that response collectively is referred to as inflammation. The word inflammation means to catch on fire. Obviously, an immune response doesn't cause you to catch on fire, but the area that's inflamed often feels hot. It feels warm because of certain cytokines and interleukins that the cells in the area are releasing. So part of the immune response is for mast cells to release histamines, and that the mast cells will sometimes release their histamines in response to things that are called superantigens. An antigen is a foreign protein. So there are some things that we call superantigens. Pollen from plants is an example of a superantigen. The pollen causes the mast cells to release their histamine, causing the inflammatory response, even though there's really no reason to, because pollen is really not so bad. It's just that mast cells happen to have specifically mast cells, overreact to things like pollen and release their histamines, and they release their histamines in the area where they encountered the pollen, which is usually in your nose. So you inhale a pollen tissue up your nose, the mast cells in the area, in the blood in that area, will release their histamines. The capillaries in the soft tissue surrounding your nose will become leaky in order to let the white blood cells out of the capillaries to get at the bad guys. The capillaries become leaky, the red and white blood cells go outside, and the plasma leaks outside, and you get swelling and redness, and that's what we call hay fever. Okay, so remember that mast cells are one of the granulocytes which is not a phagocyte. It's not classified as a phagocyte, and it is involved in the inflammatory response. And it is involved in the inflammatory response because it releases histamines, which have the effect of making the capillaries leaky. This is when granulocytes overreact to things, when they go too far. As I said, the granulocytes are part of the innate immune system. The innate immune system is kind of like the riot police. They have a habit of, their job is to clear criminals out of the area, even if those criminals are inside a crowd of law-abiding citizens. So their job is to be scary and aggressive, and so they sometimes go too far, as we all know. Okay, so the four characteristics of inflammation are redness, heat, swelling, and pain. So if you've ever had a part of your body inflamed, you know that it swells up, it turns red, it hurts, and it feels hot. And so those are the four characteristics of inflammation. So this is caused by, among other things, the granulocytes, specifically the mast cells, releasing histamine and another chemical called heparin. This allows the capillaries to become leaky or permeable. Permeable means leaky, and the blood vessels dilate, and so all of this blood plasma, red blood cells, white blood cells, will leak out of the capillaries into the area where the histamine and the heparin has been released. Okay, now what's the difference between chronic and acute? An acute illness is one that you get over quickly. It may be a bad illness, it may be a dramatic illness, but you get over it quickly. You get well quickly. Chronic means long-term. So generally when you have inflammation, it is acute. There's an acute inflammation, so it swells up, turns red, and hurts, and then eventually it goes down again. It recovers. The tissue that's affected will recover in a few days at most. But there are unfortunately, there's sometimes where a part of your body for various reasons becomes chronically inflamed. And so these granulocyte cells are always, the riot police are always there damaging the tissues. And so that causes scarring to form, scar tissue to form, and when that happens, that's called fibrosis. So fibrosis is scarring where you have, normally an organ or a tissue is made out of healthy cells, lots of them, lots of cells. If you have chronic inflammation in the area, a lot of those cells get killed and they get replaced by scar tissue, which is mostly extracellular matrix. It's mostly connective tissue and extracellular matrix. So if you have chronic inflammation of the liver, for instance, people who drink a lot of alcohol sometimes get inflammation of the liver. Eventually, that chronic inflammation of the liver will cause the hepatocytes, a lot of the hepatocytes, to die and be replaced by scar tissue. When that happens, that's called cirrhosis of the liver. And the liver becomes less and less efficient. It can only filter about 50% of the poisons out of the food you eat instead of 100. And eventually your blood becomes toxic with poison, and that will kill you. And that's a result of chronic inflammation of the liver, for example. So just remember that fibrosis is a word that you describe where healthy tissue is replaced with scar tissue. Scar tissue is mostly extracellular matrix, collagen, fibrin, elastin, proteins, stretchy proteins, but not cells. So scar tissue is characterized by having only a few cells and a lot of extracellular matrix and a lot of protein connective tissue, whereas healthy tissue is characterized by a lot of healthy cells. So you go from a lot of healthy cells, some of the cells get killed off, they tend to get replaced by scar tissue, and that is called fibrosis, when you have some organ that's affected that way. All right, what causes inflammation? Inflammation can be caused by superantigens like pollen. So as I said before, a foreign protein that causes an immune reaction is called an antigen, The word antigen comes from antibody generating. Right? So an immune response that provokes the formation of antibodies is called an antigen. A superantigen is one that provokes a severe immune response, a hypersensitivity reaction. So what we have here is an example of hay fever. Somebody inhaled some pollen. The pollen hit the side of the nose. It contacted some white blood cells. The white blood cells, in particular, the mast cells, went crazy and released all of their histamine and heparin. That caused the capillaries to leak, And so you have these red swollen eyes and often a red swollen nose because red blood cells, white blood cells, and blood plasma have all leaked out into the surrounding tissue. So this is a hypersensitivity reaction, hay fever, and it results in inflammation. Insect bites can often cause inflammation, mosquito bites and other spider bites and other things like that, because the insect saliva from insects is very immunogenic. It's an antigen as well, so it provokes the same response. Insect stings cause inflammation as well. They're meant to. When a wasp stings you, it hurts, but it doesn't hurt because of the stinger. The stinger is so small that you would never feel it if not for the fact that the wasp injected you with a poison that not only causes an immune response, but also stimulates pain receptors in the skin. So the wasp injects you with a poison that actually stimulates pain receptors on the skin. Okay, and also pressure, mechanical pressure. Somebody got punched in the face, that can also cause inflammation. So all of these activities can cause inflammation. Hypersensitivity reactions by the immune system, allergenic proteins that are present in insect saliva or insect venom, and also mechanical pressure can cause inflammation. So what is a hypersensitivity reaction? It's when the inflammatory response to superantigens, the mast cells will dump their heparin and their histamine, causing this leaky capillary response. Superantigens, normally an antigen has to go through some processing before it will provoke an immune response. A superantigen is a, for whatever reason, for reasons we don't quite understand, a superantigen provokes an immune response right away, which is normally not the case. Okay, so an allergic reaction, by the way, is called a type 1 hypersensitivity reaction because it causes this effect. Okay, so mast cells release their histamines. You get a swollen eyes and swollen nasal passages. Your nose becomes swollen up and you can't breathe, and it feels like somebody's scratching the inside of your nose with sandpaper or something like that, and you're sneezing all the time. So what do you take in order to relieve the symptoms of hay fever? You take chemicals called antihistamine tablets. The antihistamine tablets prevent or discourage the mast cells from dumping their histamine into the area. All right, so as I said, if you have chronic inflammation in an area, it leads to fibrosis or scarring. Example of hay fever, hypersensitivity. Hay fever is an example. So these are two problems with the innate immune response. There's another broader problem with the more dramatic problem with the innate immune response, and that is autoimmune diseases. So autoimmune diseases are where your own white blood cells attack your own healthy tissues. So one of the characteristics of white blood cells is that they have the ability to recognize your proteins and distinguish between your proteins and somebody else's proteins. So if your immune system, if your white blood cells see somebody else's protein, they say, well, you're not supposed to be there, and they attack and they destroy. An autoimmune disease is a disease where the white blood cells have not been properly conditioned to recognize the difference between your tissues and foreign tissues, and they occasionally make a mistake and attack your own tissues. Usually chronically, usually over the long term. Okay, so two examples that we're going to learn about autoimmune diseases. We're actually going to learn three, but two of the most dramatic ones are, number one, a disease called multiple sclerosis. Okay, so I just want to mention that nerves, in order to conduct impulses, nerves are wrapped with a lipid that insulates them and allows the signal to be conducted through the nerves. And that stuff that's insulating the nerves is called myelin. It's a lipid. What happens in multiple sclerosis is that your own white blood cells attack your own myelin and do damage to your nerves. And so multiple sclerosis is one example of an autoimmune disease. Another example of an autoimmune disease, auto means yourself, right? So immune. An autoimmune disease is one where your own immune system attacks your own body's tissues. Another example of an autoimmune disease is arthritis. So arthritis is where your body's white blood cells attack the proteins that are in the joints between your bones. So what you may not know is that there's usually a little cavity, a little pocket filled with fluid in between the bones, particularly in the hands and the knees and the elbows and so on. There's a little pocket filled with fluid, and that fluid has some proteins in it, and those proteins belong to you. They're your proteins, but your white blood cells don't realize that, and so your white blood cells are constantly attacking the proteins that are in the fluid that cushions the bones of your joints. And so your joints become constantly inflamed and they swell up, and then the bones grind against each other when you move, and that causes pain. So you'll see elderly people with arthritis, you see their knuckles on their fingers are all swollen up because the proteins in the sinus fluid in between the joints has been attacked by their own white blood cells. So you get this swelling and so damage and so on. All right, let's move on now to the adaptive immune response, which is able to kill foreign invaders by producing antibodies. Okay, so as I said, an antigen is a foreign protein that provokes the formation of antibodies. So you do need to know the word antigen, and you need to know that human cells, all of our cells have antigens on the surface, but because those are our proteins, they don't provoke an antigen, they don't provoke an immune response. So we're talking only about foreign antigens. These are surfaces on the surfaces of foreign cells that identify them as being foreign cells, basically. Okay, now here's what happens in the adaptive immune response. There's a sequence of events that happens which ultimately leads to the production of antibodies that will attack one specific foreign antigen. Okay, so first you start out with a phagocyte cell, like a dendrocyte, dendritic cell in the skin, for instance, and that cell will eat a bacteria. It will then take the bacteria apart and then put some of those foreign antigens onto its own surface. And it then presents those foreign antigens to another lymphocyte called a T helper cell. Okay, let me use an example here. Let me use an analogy here. If, let's say we're talking about the police force still. So if you have a criminal who's loose and you have a piece of the criminal's shirt, and you let the police dog smell the shirt and then say, go find the criminal, right? That when you let the dog sniff the shirt and then you tell the dog to go and get the criminal, the dog will not take that instruction from anybody. It has to be the dog's handler. It has to be the dog's trainer, doesn't it? So in this case, having the foreign antigens in the system, in the bloodstream or whatever, just by themselves will not stimulate a T helper cell to do its job. The T helper cell has to be presented the foreign antigen, the criminal's shirt, by its trainer, in this case a phagocyte called a dendritic cell. Right, so the dendritic cell eats the bacteria, takes it apart, and then presents the antigen, the foreign antigen, to a T helper cell and says, go find the criminal. Except that the T helper cell is kind of like the police dog, but it doesn't go and find the criminal. What it does is it goes and finds another type of a cell called a B cell that has the ability to make an antibody that will attack that particular foreign antigen. So we have a library of B cells in our body. We have about 2.5 million different types of B cells. Each of those B cells is able to produce antibodies that will respond to one foreign antigen but not another. So B cells are very specific. They will produce antibodies but only to a specific foreign antigen. And we have about 2.5 million different types of B cells that can respond to 2.5 million different antigens. Not all of them, not all possible antigens, but just 2.5 million different types of foreign antigens. So the dendritic cell presents the antigen to the T helper cell. It becomes something called an antigen presenting cell when it does this. So an APC stands for antigen presenting cell. It's part of the adaptive immune response. So the dendritic cell eats a bacteria, it becomes an antigen presenting cell, it presents the foreign antigen that it got off the foreign invader, presents that to a T helper cell. The T helper cell then goes and finds a specific B cell that is capable of making antibodies that will attack that foreign antigen. And then the T helper cell releases certain interleukins that cause the B cell to divide, make multiple copies of itself, and then turn into or differentiate into a related type of cell called a plasma cell. And it's the plasma cells that release the antibodies into the blood or into the lymphatic fluid to attack the invader. Okay, so a dendritic cell eats a foreign invader, takes apart the antigens, presents one of the antigens to a T helper cell. T helper cell then goes and finds one out of the 2.5 million types of B cells that we have that is capable of producing an antibody that will attack that antigen, and then the T cell makes that B cell multiply, so there's lots of them, and then differentiate into a plasma cell. The difference between a plasma cell and a B cell is that the plasma cell has a whole lot of rough endoplasmic reticulum on the inside, much more than a B cell. Do you remember the purpose of the rough endoplasmic reticulum? The purpose of the rough endoplasmic reticulum is to create proteins that are meant to go outside the cell, proteins for export. Antibodies are made of protein. So if you're going to produce a lot of antibodies, which is a lot of protein, you have to have a lot of endoplasmic reticulum on the inside of on the cell cytoplasm. So that's the difference between a B cell and a plasma cell. It's the same cell, except that it has a lot more endoplasmic reticulum on the inside because it's preparing itself to produce a lot of antibodies. Okay, so the plasma cell is what secretes the antibodies. Okay, the T helper cell is actually able to differentiate between our cells and foreign cells, foreign antigens, and they learn how to do this in an organ called the thymus. The thymus. So the T cells and B cells are both initially produced in the bone, but in order for the T cells to do their job, they have to pass through the thymus, which is sort of like a college, where they learn how to differentiate, to distinguish between our cells and foreign cells. They learn how to distinguish between our proteins, your proteins, and foreign proteins. Okay, so that is actually where the name T cell comes from, T helper cell comes from, because they had to pass through the thymus before they could become fully qualified T helper cells. Okay, now what do the antibodies do once they find these foreign invaders? Well, the antibodies coat the foreign invader. In this case, it's a bacteria. When the antibodies coat the foreign invader, that is a word called opsonization. So you need to know that word. All right, next. Once these antibodies are stuck to the outside of the foreign invaders, the liver has produced a bunch of proteins that are called complement proteins. The complement proteins bind to the antibodies, which are now stuck to the invader and break holes in the plasma membrane of the invading microorganism. Right? So antibodies in and of themselves don't actually damage the foreign invader, but what the antibodies do is they allow the complement proteins to stick to the foreign invader, and it's actually the complement proteins that destroy the invader. Okay, so this long, complicated process that I just explained to you is called a primary immune response. Phagocyte eats the invader, presents the antigen to a T helper cell. T helper cell goes, finds a B cell, stimulates the appropriate B cell to multiply and differentiate into plasma cells, and then you get antibodies produced. The antibodies attach to the invader, and then complement proteins attach to the antibodies that are attached to the invader and destroy it. That's called a primary immune response. It's usually fairly weak and does not produce a lot of antibodies, and it takes 2 weeks. That's a long time, right? That's a long time to be sick before your immune system comes to the rescue. But luckily, once it's cleared, once your immune system has done this and eventually cleared away and killed all the invaders, it leaves some cells called memory cells around that are called memory B cells and memory T helper cells that will recognize that foreign invader so that they're ready for it next time. So you don't have to go through this whole process of finding the right B cell, getting the B cell to differentiate into plasma cells. You don't have to go through that whole process again. This time we were already prepared, right? So the primary immune response is weak. It does not produce many antibodies, and it takes about 2 weeks. The secondary immune response, which is what your immune system does the second time you see the invader, the secondary immune response produces 1,000 times more antibodies on the same day, on the first day that you see it. Right, so that's why the secondary immune response is much more powerful and much more effective than the primary immune response. So I could on a test ask you to tell me, on a test I'll ask you to tell me the difference between a primary and a secondary immune response. Those are some of the things you could talk about. Okay, so you know that this whole process is coordinated by the T cells. It's the T cells that read the signal that is being presented to them by the antigen presenting cell, and it's the T cells that go and find an appropriate B cell that can produce the right antibodies. So T cells are very critical to the immune response. So a T cell is capable of telling your proteins from foreign proteins, and a T helper cell is taught how to do this. It's an analogy to say that they're taught how to do this, but they learn how to do this in the thymus. You don't need to know this, and I don't want to confuse you, but in reality what happens in the thymus is that the T helper cells are any T helper cells that do not know the difference between your proteins and foreign proteins are destroyed in the thymus, right? So that's kind of like, so then the only T helper cells that survive that process are the ones that do not react to your proteins. And so autoimmune diseases like multiple sclerosis and arthritis usually occur because that didn't happen properly in the thymus. So some T helper cells got through that still incorrectly, erroneously attack your proteins, thinking that they're invading proteins. Right, so we use the analogy saying that T helper cells learn how to distinguish between your proteins and foreign proteins. They learn how to do that in the thymus, but they don't really learn. Basically, anybody that doesn't know it from the beginning is destroyed, and then you're left with a population of T helper cells that does know the difference. Okay, now here's something that's interesting. The HIV virus that causes AIDS, the acquired immune deficiency syndrome, invades and destroys the T helper cells. And so it destroys the cells that coordinate the immune system response. So unfortunate people who have AIDS often die of diseases that other people would not die of because their immune system has been destroyed. It's been coordinated. So in other words, the HIV virus specifically invades T helper cells. That's sort of like you have an army and you kill the generals, and then the army doesn't know what to do. And so that's exactly what happens to the human immune system if the HIV virus invades and damages a large number of the T helper cells. All right, so the thymus, that is the area where the T helper cells are sifted so that you're only left, ideally you should only be left with T helper cells that know the difference between your proteins and foreign proteins. Okay, you have a repertoire of B cells. You have a library, if you will, of B cells that are capable of producing antibodies that will attach to about 2.5 million different types of foreign antigens, but not an indefinite number. Okay, so we have a pre-existing library of 2.5 million different types of B cells that can make antibodies against 2.5 million different antigens, but not for every possible antigen. Right, so if the T helper cell can't find a B cell that can attach to exactly that foreign protein, it will look for a B cell that can make an antibody that binds to a similar protein. It'll pick the closest one, in other words. And unfortunately, the closest one might be a protein that looks very similar to one of your proteins. So we call that, when that happens, we call that cross reaction. You say that you're invaded by Streptococcus pyogenes. Streptococcus pyogenes produces a protein called the M protein on its surface. We don't actually have a B cell that can attack and bind to the M protein alone, but we do have an antibody that can attack and attach to the M protein on the bacteria, and that M protein on the bacteria looks very similar to the myocardial protein of the heart. Right? So you have a cross reaction between the antibodies that are trying to kill the bacteria, and the same antibodies are doing damage to the heart. That's what happens in a disease called scarlet fever, and I'll tell you about that in a minute as well. So it's an unfortunate side effect that sometimes the antibodies that are produced to react to and destroy foreign invaders will actually react to and destroy your tissues, because the antibodies will cross-react with more than one protein. What is a vaccine? Okay, so I think we know this by now. So you get a bunch of bacteria or viruses and you kill them, and then you inject the dead bacteria or the dead viruses into a person's bloodstream, or actually you inject it into the muscle tissue, because the immune system works better if the foreign invader is not in the blood. It works better if the foreign invader is embedded in a tissue someplace. Okay, so these bacteria and these viruses are dead, so they can't hurt you, but your immune system will go ahead and mount a primary immune response against these foreign proteins anyway. And at the end, they will leave memory cells behind, so that the next time you see that invader, the immune system will spring into action immediately the same day and produce 1,000 times more antibodies than it did the first time and kill the foreign invader instantly. And that's what a vaccine is. You get a virus or a bacteria, you inject it, your immune system mounts a primary immune response. Then the next time you see the same microbe for real and it's alive, your immune system will attack it with a secondary immune response, which is much stronger and much more effective. Okay, this little interesting thing up here, this point at the top of the list, there's been some changes to that since I last made this slideshow, and obviously you might have heard of some of these changes. They are called mRNA vaccines. Okay, so you know what mRNA is, right? It's messenger RNA. So you make a copy of a gene in the nucleus, you make it out of RNA instead of DNA. You send it out into the cytoplasm, where the ribosomes will build the protein based on the messenger RNA. Okay, so you know that two of the COVID-19 vaccines, the Pfizer vaccine and the Moderna vaccine, are what are called mRNA vaccines. So this time, instead of getting the COVID, the other ones, like the AstraZeneca, for instance, they grew up a bunch of the COVID-19 virus, the coronavirus, and they killed it either by heating it up or by boiling it or by mixing it with formaldehyde. So they kill a bunch of these viruses, and then they inject you with the dead virus. That's the standard old-fashioned way of making a vaccine. The modern way of making a vaccine is you don't inject the viral proteins, you don't inject dead viruses, you don't even inject the proteins from the virus. You simply inject messenger RNA that codes for some of the proteins, and you let your own body make those proteins. Right, so that's actually much more elegant. That's actually a much simpler, much more effective way to get the foreign antigens into your body than injecting you with the foreign antigens. So instead, they inject you with the messenger RNA that codes for the foreign antigens on the coronavirus, so they don't have to inject you with the actual coronavirus, dead or otherwise. Okay, so there are a lot of different types of vaccines. There are disabled vaccines. That's where you take a virus that's very pathogenic and you mutate it so it's harmless, and then you inject that into people, so that you will then make an antibody that will cross-react with both the harmless version of the virus and the lethal version. Right now we're wondering about, because the coronavirus is mutating, the COVID-19 is mutating, we have this issue that we don't know whether the antibodies that we raised against the vaccine in the first place will also kill the variant, the mutant variant. We don't really know that. But anyway, so the oldest method of making a vaccine was to take bacteria, pathogenic bacteria or fungus or viruses, grow it up in a large quantity, kill it, and then inject it into people. The dead virus or the dead bacteria can't hurt you, but you mount a primary immune response anyway, and then you're ready for it when you see it for real. All right, so this is a graph that shows how many antibodies per microliter of blood you produce in the primary response. So you inject somebody with a dead virus here on day 1, and then eventually you see that over 2 weeks, the amount of antibodies that are produced in response to that reaches a peak in about 2 weeks. And then after the invader is destroyed, the production of antibodies goes down again. If you then inject the same person a second time, boom, right? A giant peak of antibodies is produced that destroys the invader right away. And notice the scale of this graph. Notice that over here, this peak is at 10 to the 1, and this peak is at 10 to the 4. That means that this is 1,000 times more than this. Okay, so that is how a vaccine works. And then if you take another virus and you inject it into somebody, you get a primary response, not a secondary response. That's called an experimental control. That is that part of the experiment, the red line with antibodies to B. So you inject virus A into a person on day 1. They mount a primary immune response and then a secondary immune response. They're showing you that the response is specific to the virus that you injected, because if you inject a different kind of virus, a totally different virus, you don't get a secondary response, you get a primary response, because it's the first time your body has seen virus B. Right, so that in the secondary response, this is the second time your body saw virus A, so you get a secondary response. But if you inject a different virus, virus B, you get a primary response again. Okay, so I mentioned the fact that we only have 2.5 million different types of B cells that can respond to 2.5 million different foreign antigens. That means that we will never be able to make a vaccine to every possible virus or every possible bacteria. That's unfortunate, but that's the reality. So there are some microbial diseases that we've been trying to make vaccines for years, and we've never succeeded. Like, we've never managed to get the right combination of proteins that will provoke an immune response that will make an antibody that will kill that particular virus. Okay, so we only have 2.5 million different types of B cells. Right? So that means that we'll never be able to make a proper vaccine to every possible virus. And so some examples of success stories versus failures. One of the most successful vaccines in history was the polio vaccine, which was developed between the 1930s and the 1950s. And so polio was a terrible disease, which I'll talk about in a minute. But we now do not have to worry about polio anymore, because we invented a very successful vaccine in the 50s. And everybody in the world got the polio vaccine. So many people got the polio vaccine that viruses can't live outside of humans, right? So that if you vaccinate everybody against polio, even if a few people in the world still get polio, everybody else is immune, so it doesn't spread. And when that happens, the virus becomes extinct, and you say that the virus has been eradicated. And what happens is, as soon as you get to a point in the world where around 70% or 80% of the population has been vaccinated and they are immune to that virus, that's called herd immunity. Herd immunity, as in a herd of animals, herd immunity means the entire group of humans is mostly immune, and anybody who's not immune, anybody who gets sick, will probably not be in contact with somebody who's not immune. And so the virus can't spread. That's called herd immunity. Right, so you may hear in the news that we're working very hard to vaccinate everybody against COVID-19 so that we can reach herd immunity. As soon as we reach herd immunity, we will have turned the corner, because it's harder. If most people are immune to the virus, it's harder for people who are not immune to it to spread it. That's called herd immunity. Okay, and then eventually, if no more people with polio appear, then we say the polio is eradicated, and we don't have to vaccinate people against polio anymore. And in fact, we don't. So the World Health Organization carried out a worldwide vaccination for polio. We don't vaccinate people for polio anymore, because polio has been mostly eradicated. There's another virus, variola virus, that caused smallpox. Smallpox was a terrible disease that had a 40% mortality rate. Meant that if you got it, you had a 40% chance of dying from it. we started vaccinating people. The World Health Organization, which is a branch of the United Nations, the World Health Organization vaccinated everybody in the world against smallpox, and it was eradicated in the 70s. And so now we don't have to inject people, we don't have to vaccinate people against smallpox anymore. So that's a success. The polio vaccine was a success. Rubella is one of the diseases that children can get. It's extremely contagious. It sometimes results in deafness or blindness once in a while. It's not terribly lethal, but it does damage the nerves. And so we invented a successful rubella vaccine. Cervical cancer, cancer of the cervix, which is a type of cancer that women get, is caused by a virus called HPV-16, human papillomavirus 16. We have created a very successful vaccine to stop HPV-16. So every woman should get the HPV vaccine to prevent them from getting cervical cancer. Okay, now failures. The human herpes virus, HHV1, HHV2, and so on, the herpes viruses we've been trying for decades, but have never succeeded in making an effective vaccine against herpes. And we have been trying for decades and never succeeded in making a successful vaccine against HIV. So here are some successes, and here are some failures. Let's just take another in-depth look at the polio vaccine, because it's a fascinating story. Okay, so these are little children who are in a hospital ward for polio victims. They can't breathe because they have full-blown fulminant polio, or they have full-scale polio. Polio is a paralytic disease, so if you catch a mild case of polio, you might lose the ability to walk. If you catch a full-scale dose of polio, the muscles that allow you to breathe may stop working as well. So these are all unfortunate children. Polio used to affect thousands of children and other people every year before the vaccine, and these are the children that have been hit the hardest with polio. They can no longer breathe on their own, so they have to spend their life in these machines, which are called iron lungs. So the way the iron lung allows you to breathe is you're inside it. They lower the pressure inside it, which causes the rib cage to expand and suck air in, and then they release the vacuum and let the air back in, and then the air goes back out through elastic recoil. So they're living in a vacuum chamber that keeps evacuating and then refilling, evacuating and refilling, that allows you to breathe. I mean, that's quite horrible, isn't it? So you're living in this machine for your whole life in order to breathe. Today we've made a little bit of progress where you use ventilators, but in those days they used the iron lung, because not only did they not have a way to prevent polio, but they also didn't have a way to prevent bacterial infections. And so a ventilator, an indwelling ventilator, is where somebody has a hole drilled in their neck, and then you put in a hose that leads to the lungs. That hole could get infected, and so they didn't want to take a chance and cut a hole in your neck in the 30s and prior to the 30s, because they did not yet have antibiotics. All right, so this was a terrible disease. They didn't know it at the time, but it was usually caused by people drinking contaminated drinking water. Drinking water that was contaminated with a small amount of sewage that was from people who already had mild cases of polio. Okay, now who's this guy in the middle of this picture? I don't know if you're interested in history, you might recognize Winston Churchill. You might recognize Joseph Stalin, who was the leader of the Soviet Union during World War II. Churchill, of course, was the leader of Britain during World War II. And this guy in the middle, in case you don't know, is one of the United States' most famous presidents, Franklin Delano Roosevelt. So Roosevelt was one of the three leaders of the superpowers during World War II that defeated Japan and Germany during World War II. Now, most people didn't know this, but President Roosevelt, was a polio survivor, and he was paralyzed from the waist down. So he rode in a wheelchair. He was rarely ever photographed in a wheelchair. He would put some braces on his legs, and he would stand up sometimes with the help of other people, or he would get put into the chair the way he is in this photo on the left. But the press would never take a picture of him when he was in this helpless position of having to be moved by other people. But he was paralyzed. He was what they call a paraplegic, and he was paralyzed from the waist down. And so he had polio. He was a survivor of polio. He wasn't paralyzed to the point where he couldn't breathe, but he was paralyzed to the point where he couldn't walk. Most people don't know that because in those days the press, the newspapers used to have reverence for their leaders, and so they would go out of their way never to take a picture of them when they were in a vulnerable position. You'd never get away with that today, of course. But in those days they never took a picture of Roosevelt when he was being lifted in and out of his chair or being braced into leg braces. Okay, so Roosevelt started a charity which was called the March of Dimes. The purpose of the March of Dimes, he started this program in 1936 where everybody who could would donate a dime to vaccine research to prevent polio. And so the March of Dimes gathered millions of dollars from ordinary citizens, and they put the money into polio research. And eventually in the 50s, a man named Jonas Salk finally invented a vaccine that would prevent people from getting polio. So that's called the Salk vaccine. Later on, there was another vaccine that was produced called the Sabin vaccine. So we conquered this terrible disease that used to rob children of their lives, basically. These days, there's a movement afoot for people who kind of distrust vaccines and think that they cause autism. They're sometimes pejoratively called anti-vaxxers, people who don't like vaccinating their children. One of the things that they believe is that vaccines cause autism. They don't, and this is actually one of the problems with the internet. In the old days, the problem with information was that regular people couldn't get enough information. So people were ill-informed or misinformed because they couldn't get the accurate information. Today, we don't have that problem, but we have the opposite problem. We have the problem of people getting too much information, and a lot of it is not true. A lot of it is unreliable. A lot of it hasn't been researched properly. It's just been put on the internet. You know, on the internet, everything looks equally valuable. It takes up the same space on the computer screen. If people Google it enough, then Google will lead you to the same websites, but it may not be true. That's the problem with modern information technology today. The problem is that you don't have any trouble getting enough information. You have trouble being able to distinguish the accurate information from the inaccurate information. Right, so today we have this movement, which is sometimes called the anti-vaxxers, who are against vaccinating children. Normally children at about 1 year of age are vaccinated with the measles, mumps, rubella, varicella vaccine. So they're vaccinated against 4 different viruses that cause very contagious children's diseases. And these anti-vaxxers believe that the vaccine causes autism spectral disorders. So autism is a behavioral disease. It doesn't. In fact, it doesn't. And there's lots of scientific evidence to prove that, but how do you stop a bad idea from propagating, right? So there's a saying, you know, Sam Clement was a writer, a famous English literature writer who went by the pseudonym Mark Twain, and he was famous for saying that he said a lie gets halfway around the world before the truth even gets its boots on. And that's a funny saying, but it's kind of true, because once you have misinformation that gets out into the public, and if famous people are propagating that misinformation, it's very difficult to deal with it. So this is Jim Carrey, by the way. He's a Canadian actor that's very famous, and he's a famous anti-vaxxer. He's against vaccinating children against the MMR vaccine. Okay, so I'll show you some evidence to prove that this is actually wrong. All right, so this is a paper that was published in 2002, which was done in Denmark. Now, in Denmark, they have a public health database, which means that every time you go to the doctor, the information is sent to a common database for scientists to look at disease patterns. Of course, they remove your name and any personal identification information from it, but they collect all of the information about people who got sick and what caused them to get sick, and they have it they compile it into a central database. And what these people did was to test this theory that the MMR vaccine causes autism. They wanted to test it once and for all using large numbers of participants, large numbers of subjects. So what they did was, it turns out that they had information about all the children who were vaccinated and not vaccinated between the year 1990 and the year 2000. And they found that approximately 500,000 children were born in Denmark between 1990 and 2000. Of those 500,000 children, approximately 300,000 had the vaccine and 200,000 did not have the vaccine. And then they compared what is the percentage of children that got autism in those two groups, and it was absolutely identical. In fact, the frequency of children getting autism was slightly higher in the group that was not vaccinated. Right? So you can't really prove a negative. You can't really prove that something is untrue. That's called proving a negative if you're interested in logical theory. You can't prove a negative, but this is as close as you can get to positive proof that vaccines do not cause autism. Unfortunately, this paper isn't as well known as the Jim Carrey and people who are protesting vaccinations and so on. But it's been known for a long time that vaccines, the MMR vaccine, does not cause autism. It's not associated with autism. Okay, what are superbugs? I mentioned at the early part of the lecture that superbugs are bacteria that are resistant to antibiotics. In fact, more than one type of antibiotic. So today, if you find a bacteria that's resistant to antibiotics, you can switch to a different type of antibiotics, and that'll usually solve the problem. But now we're venturing into territory where a lot of the bacteria in the world are resistant to more than one type of antibiotic. So that's getting very dangerous. Okay, so bacteria can be killed with antibiotics, but if you use the antibiotic enough, eventually they'll fight back and they will become resistant to it. They themselves will become immune to it. Not because they make antibodies or anything. Bacteria don't have an immune system, but they become immune to it for a number of other reasons, which I won't go into in this course. Right? So bacteria can be killed by antibiotics, but they can also become resistant to antibiotics. Fungi can be killed by a few different types of chemicals, and there are a few chemicals that can kill protozoa, but they're not very good. Viruses can only be killed by vaccines. Now, a number of protists and fungi can be prevented by vaccines. So remember that a vaccine prevents you from catching the illness. It does not cure you of the illness. So once you get sick, like if you already get COVID, it's meaningless to get the vaccine because, well, you can get it and you should, but it's probably meaningless because you've already caught the disease and your immune system is busy mounting a primary immune response anyway. Okay, so vaccines are preventative, and you can actually get vaccines that will protect you against many types of bacterial infections, which is something that we should be doing more of that. But anyway, colds and influenza are caused by viruses, not bacteria, and so people should not go to the doctor and get antibiotics when they catch a cold or a flu. Right, so usually one of the strongest antibiotics that we have that we tend to give to people only as a last resort because it kind of damages the person as well as the bacteria is called methicillin. So methicillin is an antibiotic that's usually used as a last resort. There are now a ridiculous number of bacteria in the world, including Staphylococcus aureus, that are resistant to methicillin. And that's what we mean when we say methicillin-resistant Staph aureus. So methicillin is an antibiotic. SA stands for Staphylococcus aureus. So methicillin-resistant Staphylococcus aureus is on the rise. Eventually, we could get to a point where a lot of the Staphylococcus aureus in the world is resistant to every antibiotic, including methicillin, and then the human race will really be in trouble. So this is a chart showing the rise of the percentage of methicillin-resistant Staph aureus in people that were admitted to hospitals. So you can see back in 1989, probably only about 2% of the people who were infected with Staph aureus in 1989, only about 2% of those infections were infections by Staph aureus that is resistant to methicillin. Look at it in 2002. We're up to nearly 40%. I haven't seen a more recent graph, but you can see that there is a problem here. So using antibiotics and abusing antibiotics is something that should not be done. All right, let's have a brief discussion about autoimmune diseases when your body's own white blood cells attack your own tissues when they should not, often because the T helper cells have been improperly sifted in the thymus. Your own body tissues are attacked by your own immune cells. Okay. They were improperly educated in the thymus. They were improperly sorted in the thymus. Two examples, rheumatoid arthritis and multiple sclerosis. So sometimes you make antibodies to an invading protein that will cross-react with one of your own proteins. That's the case in a disease called scarlet fever, right? And that's what happens when you're invaded by a bacteria called Streptococcus pyogenes. Rheumatoid arthritis, your own white blood cells are attacking the proteins in between the joints of your own limbs and hands and fingers and so on. Okay, now multiple sclerosis is where the body's own white blood cells attack the myelin sheath that insulates the neurons that make up the nerves. Okay, so there was a famous actress named Annette Funicello who was famous when I was a child, and she was a child actress, and then she grew into a very beautiful, successful adult actress. She was in a bunch of movies in the 60s, 1960s, and she is one of the famous people that had multiple sclerosis, and this is a devastating disease. So when she was a child, she had no symptoms. When she was in her 20s, she appeared normal. And then in her 30s, the symptoms of multiple sclerosis began to appear. Right, so by the time she was in her 30s, she was walking with a cane. By the time she was in her 40s, she was in a wheelchair. And by the time she was in her 50s, she was basically completely paralyzed. So here, her husband is having to move her around and feed her and so on, and she died shortly after that, tragically. And this is the result of an immune disease where your white blood cells essentially attack your own nerves, and that's so multiple sclerosis is a very tragic example of a disease, an autoimmune disease. All right, let's briefly discuss scarlet fever. Okay, this is a disease that children have. It's called scarlet fever because if they get infected by the bacterium Streptococcus pyogenes, their tongue turns bright red, so does their throat, turns bright red like a strawberry. And the Streptococcus pyogenes has a protein in it that's called the M protein. Your body will, as soon as you get Streptococcus pyogenes, if you get scarlet fever, your body will start to make antibodies that attack the M protein as a foreign protein. But unfortunately, the M protein on the Streptococcus pyogenes has a very similar shape to myocardium, very similar shape to heart muscle. And so the antibodies that are attacking the Streptococcus pyogenes will also be attacking your heart muscle and doing damage to your heart while you're a child. And this can lead to heart problems, having a damaged heart when you're in middle age, and people dying of heart attacks when they're middle-aged as a result of having gotten scarlet fever when they were young. So the take-home message from scarlet fever is, if you have a child and the child develops a red tongue like this and a red throat, take them to the doctor immediately. If you take them to the doctor immediately, the doctor will give them antibiotics to kill the Streptococcus pyogenes before the immune system can start making antibodies that attack the M protein. Okay, so this leads to scarlet fever in the youth, leads to heart damage and a possible heart failure in middle age. The way to avoid that is to watch your children, and if they show any signs of having scarlet fever, take them to the doctor immediately, and the doctor will give them antibiotics to kill the Streptococcus pyogenes before any antibodies can damage their heart. All right, now I said at the end of this lecture we're going to talk about the lymphatic system, the network of lymph vessels that run around the body carrying a fluid called lymph. So the lymphatic system, the lymphatic system as a whole refers to the immune system. So we're talking about the bone marrow, the lymphatic vessels, the lymph nodes, and the white blood cells are collectively known as the lymphatic system or the immune system. The lymphatic system tends to refer more to this network of vessels that transports fluids around the body. And the lymphatic vessels, they also help to drain excess fluid from the interstitial spaces of the soft tissues, right? So swelling in the body is prevented in large part by the lymphatic system. So blood plasma leaks out of the capillaries into the interstitial spaces between soft tissues. Some of that plasma is taken back in by the liver producing more serum albumin, but the most of it is drained away by the lymphatic system. What does the lymphatic system do with that fluid? It puts it back into the blood at the level of the subclavian veins, right? So the lymphatic system is this network of blood vessels, network of lymphatic vessels that drains fluid from the interstitial spaces and puts it back into the blood at the subclavian veins. Okay, so all of these green lines, usually on anatomy charts, the lymphatic vessels appear as these green lines. You can see it's quite an extensive network. Now these little yellow bean-shaped things are called lymph nodes. Lymph nodes. See, lymph nodes. And their job is to filter the lymphatic fluid and look for invading microbes. So each one of these lymph nodes is filled with B cells, T cells, and macrophages. And as the fluid that has been drained from the various parts of the body passes through the lymph node, they check for invading microbes, and if they find any, they destroy them. So let's go back to the police analogy. The lymph nodes, the lymphatic vessels are like roads in the body, and the lymph nodes are like roadblocks. You know, when the police stop all the cars at a certain spot in the road and they look inside to see if there are any criminals, that's what a lymph node is. Okay, so the word lymph is actually, the lymph fluid is actually derived from the blood plasma. Right, and as I said, that elephantiasis is a disease caused by Wukariria bancrofti, a microscopic worm that is just big enough to block and clog the lymphatic vessels. Sorry, I should have warned you before showing you that picture, but this is elephantiasis, and you can see it's characterized by massive swelling, right? And that's because the lymphatic system has been blocked. So that's the worst effect of Wukariria bancrofti. Okay, and eosinophils, they dump proteases onto these microscopic worms to destroy them. So if you get bitten by a mosquito that's carrying elephantiasis, Wukariria bancrofti, if you're lucky, the eosinophils will destroy a few worms. It's only if you get bitten several times and the eosinophils become overwhelmed that you end up getting full-blown elephantiasis. Okay, this is a picture of eosinophils attacking a Wukariria bancrofti worm. Okay, so the lymph nodes are filled with phagocytes, T helper cells, and B cells, and they check for invaders, including Wukariria bancrofti, including Staph aureus and Streptococcus pyogenes. They check for these things in the lymph fluid, and if they find any, they destroy them. So they're kind of like a police roadblock that's checking the traffic as it goes through. This is something called immune surveillance, you know, where the police department, the immune system police department is surveilling the area to make sure, constantly surveilling and checking in different areas of the body. Okay, so what happens if your lymph nodes swell up? If any of you are interested in being a doctor or a nurse, you need to know this. When your body is fighting an infection, the lymph nodes that caught some of these criminals will swell up, because if they catch a criminal, if a lymph node encounters a microscopic worm or a cancer cell or a bacteria or a virus, the B cells and T cells and macrophages will multiply inside that lymph node in order to do battle with that invader. So lymph nodes will swell up if they encounter invaders. Right? So one of the easiest ways that you can check to see whether or not somebody is fighting off an infection is to feel their lymph nodes and see and feel if they're bigger. So typically when you go to a doctor for a checkup, one of the things they do, you say, I feel sick, I feel feverish or whatever, they come around and they feel under your chin. And under your chin is a cluster of lymph nodes that are called the submandibular lymph nodes. Submandibular lymph nodes right under your jaw. And then they feel your armpits, because under the armpit is called the axillary area. And so they feel there's a cluster of lymph nodes there as well, and they feel those, and those are called the axillary lymph nodes. Furthermore, when women do a breast cancer self-exam, one of the things that you do is you feel in the armpit for lumps. If you feel a lump, that lump is probably, I hope you never do, but if you do, that lump is probably a swollen lymph node. And that means that that lymph node is working hard, and the white blood cells inside the lymph node are dividing in order to fight cancer cells that are traveling out from the breast through that lymph node. So they're desperately trying to, the lymph nodes are desperately trying to stop the cancer cells from spreading. Spreading is called metastasis, by the way, metastasis. So if a cancer has spread to the various parts of the body, they say it is metastasized to the various parts of the body, which is usually very bad sign. And also the spleen is a giant lymph node that cleans microorganisms from the blood, as well as breaking down the broken down old red blood cells. Okay, so here we have the axillary lymph nodes. That's one of the places where women feel under the armpit to look for swelling as a sign of breast cancer. I hope you never feel that, but that is what you should be checking. And the submandibular lymph nodes are right under the chin, so you feel them to see if they're swollen. So if either of those places have swollen lumps, it means that your body is trying to fight off an infection. Okay, so to summarize this long and somewhat confusing lecture, we started out by talking about what kinds of things can invade your body, and then we talked about a lot of things to do with the different types of white blood cells that constitute the immune system. So we talked about the blood cell lineages. We have a myeloid lineage and a lymphoid lineage. We talked about leukocytes, the fact that leukocytes are divided into granulocytes and lymphocytes that have different jobs. Generally, the granulocytes are involved in the innate immune system and the pyogenic response, as well as inflammation. And the lymphocytes are involved with the adaptive or learned immune response, which includes a primary immune response versus a secondary immune response. And we exploit the secondary immune response when we make vaccines. And we also talked about autoimmune diseases and hypersensitivity reactions. And we also talked about the lymphatic system, referring to the lymphatic ducts and the lymph nodes. All right, so the next lecture will be on the renal system, which is on the kidneys, and I'll see you there.